Hello, everyone, and a warm welcome to this webinar co-produced by the CRISPR Journal and Genetic Engineering and Biotechnology News. Our presentation today, it's going to be a good one. It's entitled CRISPR-Based In Vivo Genome Editing and Base Editing, and it's sponsored by TriLink Biotechnologies. This is the first in a three-part series that we are producing in association with TriLink. I'm your host, Kevin Davis, Executive Editor for the CRISPR Journal, which has been publishing research in CRISPR biology and genome editing now for 2.5 years. And I'm also the Editor-at-Large for GEN, which has been covering the biotech industry for a little longer, 4 decades in fact. Let's meet our special guest speaker for today's webinar. Dr. Wen Xue is Assistant Professor in the RNA Therapeutics Institute at UMass Medical School in Worcester, Massachusetts. A little bit about Dr. Xue. He studied at Nanjing University in China before moving to the USA. He did his PhD with Scott Lowe at Stony Brook and a postdoc with Tyler Jacks, the director of the Koch Institute in Cambridge, Mass, before moving west to start his faculty position at UMass Medical School in 2014. As always, we would love to hear from you by submitting a question during the presentation that we can address at the closing portion of today's webinar. We'll try to get to as many questions as we can. So please simply hit the Ask a Question button that you'll see on your screen and hit submit. We'll gather those and we will get to them at the close. All right, let's get our webinar underway. Dr. Xue, we are so looking forward to your presentation. I'm going to hand the microphone over to you.
Thanks for the introduction and welcome to today's seminar. My lab studies CRISPR-based in vivo genome editing in mouse models. There are a large number of single-gene genetic diseases. For many, there are no effective therapy. So gene correction can provide long-term therapeutic benefits. We aim to develop a programmable platform for mutation correction in vivo. So just a brief introduction of the CRISPR system. The Cas9 protein and the sgRNA will bind to the genomic target and create double-stranded DNA breaks. These breaks will be repaired by either NHEJ pathway leading to indel formation or by homology-directed repair or HDR pathway using template DNA, and this can mediate precise gene editing. There are newer versions of the CRISPR effectors, such as the base editor and prime editors developed by David Liu's group. The major challenge for CRISPR-mediated gene correction is delivery. So for gene correction through HDR, we need to deliver 3 modules. First, Cas9, second, guide RNA, and the third, repair template DNA into the mammalian cells. And we have developed nanoparticles to deliver the Cas9 mRNA and the adeno-associated virus, AAV, to deliver the guide RNA and repair template. As a proof of concept, we studied in vivo CRISPR delivery in a mouse model of tyrosinemia. So this is a metabolic liver disease. The mutation is in the gene called FAH, which is a key enzyme in the tyrosine catabolic pathway. FH mutations will cause loss of function, leading to a toxin buildup in hepatocytes and apoptosis. This can cause severe liver damage in patients. It's a rare disease, and the current standard of care is to restrict tyrosine in the diet. And the patients have to take lifelong treatment of an inhibitor called NTBC, which blocks the catabolic pathway. And this can increase long-term liver cancer risk. And we're using a mouse model created by Marcus Grumpi's lab. This is a G2A point mutation at the last nucleotide of exon 8. This mutation causes exon skipping of the exon 8, leading to loss of function. To deliver CRISPR components in mouse liver, we collaborated with the Anderson's group to investigate Cas9 mRNA delivery by nanoparticles. We used TriLink's chemically modified Cas9 mRNA, and Dan's group has developed liver-targeted lipid nanoparticles. So we take wild-type mice and inject Cas9 mRNA nanoparticles by tailvein injection. By qPCR, we can detect Cas9 mRNA at early time points, and it's rapidly degraded by 24 hours. We also used beta-gal as a reporter gene and delivered its mRNA in nanoparticles. By staining liver sections, we can detect very robust beta-gal signal post-injection. And here is a slide kindly shared by Anton McCaffrey at TriLink, showing the CleanCap mRNA used in our in vivo study. So this platform simplifies mRNA manufacturing, improves yield, and also allows GMP manufacturing. Because mRNA expression is transient, we hypothesize that Cas9 mRNA may reduce off-target genome editing. We chose an sgRNA targeting VEGF-A with high off-target effects, and we compare either lentiviral Cas9 or the Cas9 mRNA. By measuring the off-target to on-target ratio, we show that the Cas9 mRNA has a much lower off-target rate. This is a major potential benefit of delivering Cas9 using a transient delivery method. In addition to the Cas9 delivery module, we used AAV to co-deliver the guide RNA and HDR template. So the template DNA harbors homologous arms and the correct gene nucleotide. So in the FH mutant mice, we first injected AAV to allow guide RNA expression. And 1 week later, we dosed by tailvein injection the nanoparticle Cas9 mRNA. We kept these mice on NTBC to measure the initial gene correction efficiency. So by staining the liver sections using FH antibody, we can detect FH positive cells in the mice injected with this combination therapy. One single treatment can generate up to 6% of FH positive hepatocytes. And one phenotype of these FH mutant mice is that all the control mice will rapidly lose weight when we removed NTBC water. So compared to these controls, this red line shows that the treated mice now show a full stabilization of their body weight. So they are cured of this FH body-mediated weight loss phenotype. We also show that by guide-seq there is low off-target effect using this mRNA delivery method. And there is no detectable liver damage markers such as AST or ART using AAV and nanoparticles. So this study shows the potential of gene correction using CRISPR HDR method. So far, we showed HDR gene correction for one particular FH mutation. In patients, there are many types of loss-of-function mutations throughout the coding region. And this is a common feature for many types of recessive genetic disease. So for the patient population, we would need a large number of tailored HDR template and guide RNA pairs. So many FH patients harbor compound heterozygous mutations. Namely, the patient has two types of mutations on both chromosomes. We hypothesize that if we use a guide RNA to cleave between the mutations, some cells may undergo allelic exchange by interchromosomal DNA ligation. And this can create a copy of wild-type functional allele without the need for DNA template. To test this idea, we first created a compound het FH mouse model. So we crossed two different types of loss-of-function alleles. One harbors a neomycin insertion in exon 5, and the other one has the same exon 8 splicing mutation. We cloned the guide RNA targeting the intron region. In these mice, we injected AAV carrying Cas9 and the intronic guide RNA. And by staining liver sections, we were very excited to detect FH positive hepatocytes clones. suggesting that allelic exchange approach can generate functional cells in vivo. There are several potential advantages of allelic exchange compared with traditional HDR method. First, there is no need to deliver DNA template, simplifying the design of delivery vehicles. Second, the DNA repair is mediated by NHEJ pathway, which can happen in non-dividing cells such as neuron or muscle cells. Third, the target can be chosen in the intronic region, so we have flexibility in selected target guide RNA, and these intronic India mutations are less likely to be detrimental than the exon-targeted guide RNAs. And the most interesting feature of this method is that one guide RNA can potentially work for several types of compound head mutations, as long as the guide RNA target is between the two types of mutations. Several technical caveats need to be considered. Because this is intrachromosomal DNA repair, the efficiency is relatively low. Fortunately for the FH mouse model, the corrected cells will have a proliferation advantage, so this is sufficient to rescue the phenotype in mice. And of course, this method does not work for homozygous mutations. To address these issues, we want to study another type of HDR independent gene correction method, which is base editing. So recently, the base editors, such as adenine base editors, are developed for correction of single nucleotide mutations, and this is developed by David Liu's group. So the ABE is a fusion of the Cas9 nuclease with evolved E. coli TED-AD aminase. And this complex can convert an AT base pair into a GC base pair within an editing window, usually from position 4 to 9 in the guide RNA region. So this is a promising gene correction method because around 50% of mutations are G2A mutations. There are studies showing off-target DNA and RNA editing, but there are newer versions of ABE that can be engineered to reduce such off-target editing effects. So our FH mutant mouse has a G2A mutation, making it a good candidate to test ABE in vivo. So here is the sequence of the FH mutation. We identified a TGG PAM sequence, and using this guide RNA, this red mutation A will be at the position 9 of the guide region. And there's another A6 within the editing window, and this bystander editing can create a codon change. I'll come back to this in a minute. So we first tested base editing in vitro using MAF cells from FH mice. So we co-transfected this guide RNA with two versions of ABE. And we show that ABE 6.3 has a higher level of A9 base editing compared to the 7.10 version. To deliver the ABE in vivo in the FH mice, we use a technique called hydrodynamic tailwind injection. This can deliver plasmid DNA, both the guide and ABE, into a fraction of hepatocytes for transient expression. Again, we use the body weight as a primary readout of the mouse phenotype. So the PBS control and the ABE alone control rapidly lose body weight. In contrast, the ABE and guide RNA treated mice showed a full recovery of body weight at 1 month post-NTBC water withdrawal. At the endpoint, we analyzed these mouse livers by IHC staining, and we can detect the clones of FH-positive hepatocytes in the ABE-treated animals. Because this FH mutation affects the splicing of exon 8, we performed RT-PCR in liver lysates. So the mutant FH mRNA shows a truncated band lacking exon 8. And in the ABE-treated livers, now we can detect the full-length cDNA band containing this exon. These data indicate the restoration of FH splicing by ABE treatment. We also analyze the DNA level by deep sequencing, and we can detect this on-target A9G editing and some level of A6G bystander editing as well. So our in vivo study demonstrates the efficacy of ABE in an adult mouse model. During this study, we observed that original ABE is not very efficient in cells. So we collaborated with David Liu and Lucas Dow's groups and optimized the original ABE. So we codon optimized the Cas9 nickase, we removed the potential polyA sites in Cas9, and we also added an additional N-terminal NLS sequence. So we call this optimized ABE as reassembled ABE or RA6.3. So when we transfect the 293 cells with two versions of ABE plasmids, we can detect a higher protein expression of RA6.3. As a functional readout, we chose an endogenous genomic target with multiple A's. And by deep sequencing, we show that this improved RE6.3 shows higher editing efficiency compared to the original version. And this is also true for two additional target sites in 293 cells. Next, we compare the base editing efficiency of RE6.3 with the original ABE in the same mouse model carrying FH mutation. So using the FH-positive hepatocytes as a readout of the editing efficiency, we show that this RE6.3 has improved editing efficiency in vivo, just like the in vitro 293 cell data. So together, our optimized version of RE6.3 provides a powerful genome editing platform to mediate in vivo genome editing. A quick summary here, we show that both HDR and ABE can be applied to correct mutations in vivo in a mouse model of human disease. Compared with HDR, ABE has advantages such as there's no need for DNA repair template, and ABE also generates only low levels of India mutations. There are still limitations of current ABE tools. First, because of the editing window, sometimes it's difficult to find the PAM sequence to target a mutation. The newer versions of Cas9 with more flexible PAM sequences, this can help to address this point. Second, there are often bystander A's within the editing window. Sometimes they will create missense mutations by the standard editing. So this needs to be studied in a functional assay. And finally, because ABE is even bigger than Cas9, it's very difficult for delivery in vivo. Several groups have developed a split inting-based approach to deliver ABE using an adeno-associated virus approach. So we want to test whether ABE can be delivered as mRNA. There are potential advantages of mRNA delivery. The first is a transient expression, which can lower the off-target effects, and this can be applied to edit difficult to transfect cell types, can potentially reduce the risk of vector integration. And finally, mRNA can be used for repeated dosing in vivo, which is very challenging for viral vectors due to the capsid immune response. So we started with our ABE RE6.3. And we already showed that using plasmid transfection, this version of ABE can produce robust protein expression in cultured cells. So Anton TriLink helped us to synthesize several versions of mRNA, either the unmodified wild-type mRNA, the sequence optimized through uridine depletion, and a chemically modified 5-MOU version in addition to the U depletion. Tintin Zhang, a postdoc in my group, tested these mRNAs in 293 cells. So we observed that the wild-type version of mRNA is poorly expressed in cells. Importantly, this 5-MOU and U depletion version shows a robust protein expression. It's comparable with Cas9 mRNA. So Tintin also tested guide RNA chemical modification. So when we combine with modified ABE mRNA with unmodified native guide, we detected a very low level of editing in cells. Inspired by the work by Metapotus and others, we chose a chemically modified version of guide RNA with a 5' and 3' nucleotides with chemical modifications. Using these modification combinations, now we can detect a high level of base editing in cell culture. As a proof of concept, we chose to target a CFTR mutation in lung airway epithelial cells. So the cystic fibrosis is caused by mutations by CFTR, which is a chloride channel. For some missense mutations, there are FDA-approved small molecule drugs modulating the channel activity. Unfortunately, for patients with nonsense mutations, there are no approved therapies available. We chose to target a common CFTR nonsense mutation, the W1282X. So this is a G2A mutation resulting in a premature stop codon. So there is an isogenic cell line model of this mutation using human lung airway epithelial cells. So though the mutant cells do not have chloride channel activity compared to the wild type, recapitulating the phenotype of primary CF patient cells. A technical difficulty is that this cell line is not amenable for GFP plasmid transfection. Fortunately, we can electroporate GFP mRNA and detected robust GFP expression. So we chose this cell line to test our ABE mRNA delivery in vitro. So we cloned guide RNA with this TGG PAM. Within the editing window, the mutation A in red is at position 9. In addition, there's a bystander A5, and this editing will change the codon of CFTR. So we electroporated both ABE mRNA and this guide RNA into the mutant cell model. By deep sequencing, we indeed detect both the on-target editing in red and the double editing in blue. To study the function of these alleles, we isolated clones representing these two editing genotypes. And in collaboration with Martin Mans and the CF Foundation Therapeutic Labs, we performed electrophysiology to study the function of CFTR in these clones. On the right shows the CFTR electrophysiology assay results. The top panel shows the wild type and the mutant parental cells. On the bottom panel, we see the CFTR chloride channel activity is restored in both cell clones, suggesting that base editing can restore the function of this CFTR mutant. And importantly, this particular mutant allele caused by bystander editing does not affect the CFTR function. In summary, we have developed an optimized mRNA modification platform for ABE. We showed that chemical modification to both ABE mRNA and guide RNA enables efficient base editing in lung epithelial cells. Our data show that ABE corrected CFTR mutation and restored electrophysiology in cultured cells. And finally, our results highlight the need to study bystander editing in a functional assay. To explore the CRISPR delivery in the lung, we collaborated with Gongping Gao's group to study AAV-mediated genome editing in lung epithelial cells. So we used an AI9 reporter mouse. So the mouse has a TD tomato allele silenced by an upstream stop cassette. We used 2 guide RNAs to excise this stop, allowing tomato to report genome editing events. So we cloned 2 AAV vectors, one encoding the SpikeCas9 sequence, and the second encoding 2 guide RNAs together with a GIP reporter. And delivered to the mouse lung, we performed intranasal injection using the AAV serotype 5. And when we perform IHC staining in lung sections, in dual AAV group, we can detect GIP expression in lung epithelial cells. Importantly, we also detect tomato positive cells in the lung airway. We can achieve up to 12% using AAV injection platform. Finally, I'd like to include 2 slides from Anton. So our preclinical study using Cas9 and ABE has tremendously benefited from TriLink's research-grade RNA reagents. TriLink also offers flexible scale-up and early-phase GMP manufacturing to support clinical studies, including vaccine development. In the Q&A session, Anton can give you more details regarding TriLink's expanded mRNA manufacturing capacity to meet the global pandemic need. I will end the seminar by acknowledging the lab members in my group. So the CRISPR and base editing work is led by two postdoc researchers, Tingting Zhang and Chengqing Song. So I want to thank my collaborators and colleagues. So I am in the RNA Therapeutics Institute at UMass. So we have the great RNA researchers including Phil Seymour, Craig Mello, and Eric Songheimer, and others. So this is an incredible environment to do cutting-edge research on RNA biology and therapeutics. So the CRISPR work is in close collaboration with Dan Anderson, David Liu, and Hao Ying's groups. Martin Mann's and Hilary Valley's group at the CF Foundation Therapeutics Labs have provided critical help on the cell line model. And Anton's group at TriLink has provided valuable reagents and advice to support our RNA research. And we were also very grateful to the funding agency, including NIH, American Cancer Society, and CF Foundation for supporting our research. I will stop here and thank you for your time.
Wow, thank you very much, Dr. Xue. That was an excellent presentation. We are still willing to accept your questions for the Q&A portion coming up, so please send them in using the Ask a Question button. A quick reminder that we will be archiving today's webinar. You'll be able to view it for 12 months on demand, or share the links with your colleagues, and we'll get you that information after the broadcast. All right, I think it's time then for us to move into the Q&A sessions. The questions are coming in, so let's recall our speaker. Just give us a couple of moments to transition into the live Q&A portion of the program.
Okay, we're live, and in addition to our guest speaker, Dr. Xue, we're joined by Anton McCaffrey, sitting in as a special guest. Anton is Senior Director of Emerging Science and Innovation at TriLink, and will be able to add his expertise to certainly for some of the questions which have been flooding in. And we have a good deal of time, which is rare for these webinars. So let me just pull up the questions, and we will get stuck in and keep them coming, and we'll try to get to, we've got probably a dozen in the queue, so let's get on with it. First question, great presentation, Dr. Xue. The approaches you described utilize AAV or mRNA-based delivery of the guide RNA. Have you used synthetic guide RNA for in vivo editing, and how does this approach compare to other in vivo editing approaches?
A great question. So the answer is we have used the synthetic guide RNA. And so in another collaboration with Dr. Dan Anderson and Hao Ying, we have tried chemically modified synthetic guide RNA in combination with Cas9 mRNA. And specifically, we target a gene called PCSK9, and we can achieve more than 80% indels or gene knockout in the liver using this delivery method. Yeah. And Anton, do you want to chime in for the synthetic guide and the chemistry available at TriLink?
Sure. So there's a variety of different chemistries that are available. The ones that are most common were based on some work done by Matt Porteous's group at Stanford. So those involve having 3 phosphorothioates on either end and 3 2'-methyl groups. But there's also some very interesting work that Wen could probably comment on from the Sondheim group at UMass, where they modified most of the bases within the synthetic guide to make it as chemically stable as possible. And I believe that was published in a paper about a year ago. I don't know if you have more comments on that one.
Yeah, and my colleague at UMass, Eric Songheimer, anesthesia, and others have shown that there is a possibility to heavily or near completely modify the guide RNA sequence, which is 100 nucleotide in length. And so there are still ongoing work trying to using chemical modification to stabilize guide RNA in order to facilitate in vivo delivery.
Okay, thank you both. Let's move on. How many genetic disorders are there with single base mutations, Wen?
Yeah, it's another great question. So there are many genetic disorders with mutations. So that creates a very daunting task for researchers to develop therapy or gene therapy for each individual disease type. So the beauty of CRISPR-Cas9 is it's very flexible to manipulate genome. So we can develop guide RNAs, and it's very user-friendly. So we can very flexibly target mutations you're interested to study.
Okay, great. Next question. How do you make sure that the Cas9 mRNA escapes endosomal entrapment within liver cells?
Yeah. So this we as I mentioned in the talk, we deliver the Cas9 mRNA using the lipid nanoparticles developed in Dan Anderson's group at MIT. So the trick is the lipid nanoparticle formulation. So Dan's group has screened large libraries of lipid and other chemical groups, and eventually they have identified nanoparticles mimicking the natural lipids. So by taking advantage of the LDLR receptor and pathways, these nanoparticles can, mimicking the natural lipid metabolism, they do escape endosomal trapping and then release the mRNA cargo into the liver cells. That's how we detect the beta-gal staining and also the Cas9 functional readout by the genome editing events.
Okay, very good. Next question, did you monitor potential RAAV8 integration at the CRISPR-Cas9 cut site?
Yeah, great question. So there are papers reporting an AAV or partially the AAV-ITR sequence integration at the cut site. So there are studies from Charlie Gersbach at Duke and other groups showing this potential caveat or side effects of Cas9 cutting and gene repair events. So we haven't done deep sequencing in the studies I have presented, but we are actively monitoring an AAV integration. And as I mentioned, one motivation for us to develop an RNA-based delivery is that to reduce the risk of factor integration. So by using mRNA and synthetic guides for in vivo, we can reduce or eliminate viral vehicle delivery.
Okay. How long do the homology arms need to be? Can this approach be used to insert genes, for example, Cre recombinase or GFP?
Yeah, and the short answer is yes. So the HDR has been widely used to insert genes such as Cre or fluorescent reporters into the genome, and alternatively to incorporate and rescue genes into the safe harbor genomic locus described by many groups. So in general, we use homologous arms close to 800 base pair. So the papers are using either from 200 base pair to even longer to kilobase scale, depends on the cell type or how efficient the homologous recombination is. And another consideration is the cargo size. For large gene insertions, we need longer arms to increase the HDR rate. So this is another great question.
Okay. The next question, I think, is looking at COVID-19, but I think in a therapeutic sense rather than a diagnostic sense. How are CRISPR and Cas9 associated with coronavirus?
Kevin, so as you alluded to, many ways to think about this question. So as a biological question, the CRISPR and coronavirus are unrelated. We know RNA virus has very small genome and it's all RNA encoded. So coronavirus doesn't have any CRISPR or Cas9 because it's all DNA encoded CRISPR enzymes. As a biotech or detection question, so there are many exciting researchers ongoing trying to develop CRISPR-based viral detection methods. Or Standard Tree group show that Cas13 and RNA-guided CRISPR can be potentially utilized to knock down or silence influenza or similar RNA virus. So in short, CRISPR can be potentially utilized to combat this virus infection or to facilitate detection.
So this is Anton.
Go ahead, Anton.
I think it's interesting. Yeah, I think it's interesting also to mention. I know you asked Wen to address the therapeutic side rather than the diagnostic side, but I think that there's some very interesting developments coming out of Feng Zheng's lab in Cambridge and Jennifer Doudna's group using this as a diagnostic test. So Cas13 uses a guide to recognize an RNA, but once it recognizes that RNA, it now degrades other RNAs around it. And so there's been some very exciting developments from a company called Sherlock and another called Mammoth, where they have used this to develop a point of service type of diagnostic that happens very rapidly, where basically you would put in a sample from someone who's infected. If it contains a COVID RNA, then basically it activates the Cas9 variant to degrade other RNAs in the sample, which can be, a TACMAN probe, for example, and it gives you a color readout of whether or not the person has COVID genetic sequences. And I think this is a really exciting development for the field.
Yes. There's been some, thank you very much for that, Anton. There's been some great reporting by my colleague Juliana Lemure in Genetic Engineering and Biotech News. You can find that online at the GEN website for people who are interested. I think the question may also have been perhaps alluding to the recently published work of Stanley Chee's group at Stanford, but now I think of it, that wasn't Cas9. I think that was using Cas13 as well in a Pac-Man approach to potentially destroy the COVID-19 virus. But I think in the interest of time, we will move on because we're here to talk about Wen's work, not Stanley's work. The next question is, in general, for generating point mutant cell lines, do you generally use or recommend HR, homologous recombination, or is base editing reliable enough to use instead? That's from Soren.
Yeah. So we are currently using both approaches in our hands. It really depends on the sequence context near the mutation you want to engineer. So if the base editing has a very close by PAM and the product is highly pure, meaning there are no adjacent A or Cs in the neighborhood or in the near the mutation, if there are not many high-level bystander mutations, the base editor is a very efficient tool. In the meantime, HDR has very high purity, so we can engineer a sequence of the exact mutation we want to insert. So we're using both approaches depending on the exact mutation on the sequence we want to engineer.
Okay, great. I'm rattling through these. Is hydrodynamic injection liver specific?
Yeah, so it is a liver specific or liver targeted delivery method for research purpose. So in animal models, during injection, more than 90% of the plasmid uptake is in the hepatocytes because of the property of hepatocytes to uptake vehicle-free DNA. So it is a highly liver-targeted delivery method.
Yeah. Both of you, I think it would be great. A question we had in mind before the webinar started was to discuss what is known currently about Cas9 side effects, or I guess off-target effects in vivo. What's the latest Wen, I'll start with you, and then we'll bring Anton in. What's the latest in terms of people have been worried about these off-target effects for years, but is the feeling in the field that you have this problem more or less under control, that we understand enough to minimize these in a therapeutic setting?
So great point, Kevin. So we're having much better toolkits right now to detect off-target effects. There are GuideSeq or many high-throughput genome-wide sequencing platforms developed over the years to detect off-targets. So we do have a much better means to profile, to monitor these off-target genome editing events, both for Cas9 and for base editors as well. And in the meantime, there are many Cas9 variants being developed, showing much lower off-target editing. So it is being a field heavily studied by many groups in the field. And besides the Cas9, engineering Cas9 itself, there are also studies on trying to modify guide RNA or to change the secondary structure of guide, also with the goal to mitigate off-target editing. So again, there are exciting developments in the off-target field. Besides that, Cas9 is known to trigger immune response in vivo.
Yeah.
So that's another consideration and there are many studies ongoing. The previous question mentioned an potential AAV integration at the Cas9 cut site. That's another layer of complexity of vehicle or partial sequence integration in the Cas9 cut site. So there are many things we need to learn or to optimize before this exciting CRISPR toolkit can be translated into a therapy.
Anton, did you want to add anything to that?
Sure. I think that there's been an extensive look at this in the literature and amongst the scientists that have been studying CRISPR, and I think that this differs from other technologies in the past. For example, like RNA interference, where initially people were not really looking at that very much, but from the very beginning, people have been looking for these off-target effects as running towards the fire as opposed to running away from it. And as Wen noted, there are a number of approaches in terms of modifying the guide strand or modifying the Cas in order to reduce these responses. And it also appears to be somewhat specific to different guide sequences. And one of the luxuries we have with Cas9 is that you can choose different guides and then monitor their off-target effects and look for ones that are particularly less prone to having those effects. And so I think the field has embraced the question, and I think it's something that can be addressed. And the other thing to note is that the occurrence of double-stranded breaks in cells is not an uncommon event. Actually, there's quite a few of these that occur naturally and are repaired naturally. And so many of these are probably going to be relatively benign, but I think it's something that has to be addressed as you move to the therapeutic field.
Yep. Okay, great. I've got two questions back to back here that are sort of related, Wen, to see if we can roll these into one answer. Firstly, I need to create a homozygous point mutation in the cell DNA, but I only have a heterozygous point mutation. Can I create the same point mutation on the other allele so it will become homozygous? And the companion question is, how do you generate biallelic mutations? What are your thoughts there?
Yeah, so if we use base editors, so we can definitely edit, because it's a defined base pair change, we can use base editor to target the unmodified Y-type allele. So it is feasible to engineer a heterozygous cell line to make it and select clones that are homozygous. So that's definitely doable. Yeah, and alternatively, it's also studies showing using HDR approach to edit the the locus, the genomic site. And by it's more labor intensive. We need to select clones or using different assays to enrich the homozygous allele. But the answer is yes, it's possible.
The answer is yes. Anton, anything you want to add to that?
No, I think I really don't.
Okay, no worries. We'll move right on. Another twofer. So Arnab from the Anderson Lab, thanks for viewing the webinar. Arnab asks, Wen, how do you compare different CRISPR technologies in terms of their ease of use, although they have different applications? I'm thinking of base editing, prime editing, which we haven't really talked about, and traditional genome editing with Cas9. And then Subangi asks, who's a student, asks, how is base editing beneficial over current methodologies, apart from being highly specific?
So again, great questions. So we are constantly asking the similar questions, how we compare base editing, prime editing, and Cas9. And the short answer is really depends on the project or the genome editing you want to achieve. And these are highly complementary tools. So they can complement each other to edit genome or to enrich the genome engineered events. In our hands, we are depending on the project. If we want to insert large reporters or large fragment insertions, it's only feasible with traditional Cas9. So the prime editing can insert small fragments or mediate deletions or all types of nucleotide changes. And again, for base editing, it's highly efficient for single base conversions, either ABE or CBE. So David Liu's group showed nicely that for most sequences they assayed, base editing is a little bit more efficient than prime editing. And the caveat or the beauty of prime editing is super flexible. It can mediate all types of changes.
Yes.
So in general, we are exploring all these toolkits. And a common theme is how we, for in vivo, how we deliver these toolkits into the cell type of interest. And because these are all Cas9 or Cas9 nickase-based enzymes, we're trying to develop a universal delivery vehicle such as AAV or mRNA nanoparticles to facilitate in vivo studies. Yeah.
Yes. There's been a lot of recent publications evolving new versions of base editors, in particular ABE, that are tens, if not hundreds of times more active than the originally published editors. Do you grab onto these new super active enzymes and are we done with the old original ones that were in the Nature papers? Are we now moving on to these new iterations of base editors?
We need David Liu to chime in. Just kidding. So the ABE80 recently published by David's group is definitely a powerful tool and shows very improved kinetics and much faster and more active in most sites. And so we're definitely very interested in testing this new editor or any newly evolved base editors published by the colleagues in the field.
Right. And these are, am I right in thinking that these are available through AdGene? Is that where you would advise researchers to turn to get these materials?
Yes, and AdGene is actively providing the researchers with these new toolkits.
Very good. Anton, I want to bring you in because we need to get to this question. Does TriLink manufacture GMP-grade synthetic guide RNAs?
Yes, we do. So we have a newly developed facility that we just moved into around the beginning of the year that expands our GMP capacity by fivefold. We have 5 independent suites for making messenger RNAs, GMP messenger RNAs, also GMP guide strands. So some of the guide strands that are used in base editor are easily accessible. Those are around 100. We've also been making some guide strands for prime editor. These are a little bit more challenging because they're longer, around 127 nucleotides. But absolutely, yes.
Okay. I've just looked at the questions in the queue. I mean, it's ridiculous. We had nearly 2,000 people register for today's webinar, so extraordinary interest. We're not going to get to every question, but let's take the next 10 minutes as the rapid fire round, and so we'll ask Wen and Anton for quick, short, concise answers if possible. Wen, how do you target nanoparticles to a specific tissue in vivo? That's from Zara.
So we're collaborating with Dan Anderson and his group at MIT. So Dan's group has developed different types of nanoparticles engineered to target tissue types in vivo. For example, I showed the liver-targeted nanoparticle, but Dan's group has other tissue-specific or targeted nanoparticles.
Okay. Kusali wants to know, what's the efficiency of using 2 guide RNAs to cut out the stop cassette in the mice?
So the log stop is pretty efficient if the 2 cut sites are close to each other. So it is in cell culture we can get close to 30% tomato positive cells.
Okay. How do you ensure the targeted delivery of Cas9 RNA to liver cells? I think we've really covered that one. So let's move ahead. Thanks for your presentation. What application do you have to reduce immune rejection? What's the success rate of this technique? Is there a carrier you know from the cell itself? Can phospholipid-coated transport be used? Can you respond to that?
Yeah, so regarding the Cas9 immune response, so it's an actively studied field because it's a major barrier to apply this CRISPR delivery or in vivo editing directly in patients. And in general, it's underdeveloped, so there are no currently available tools besides some work showing there are mapping of the epitopes on Cas9 from George Church's group.
All right.
Anton, could you comment on the are there like RNA in particular for RNA and mRNA modification? Are there particular more chemical modules that can help to reduce the mRNA immune response? That's another layer of immune rejection if we deliver Cas9 mRNA.
Sure. So Wen didn't get into this in a lot of detail in his talk, but one of the things that we think about a lot is that about how immunogenic the different components can be. So you have, a Cas9 mRNA, you have a guide strand, and in some applications you have a donor. The donor may be the most immunogenic part of all, the donor DNA. But in engineering this experiment, we tested different kinds of chemical modifications and sequence optimizations in order to minimize the immunogenicity of the messenger RNA. So a cell seeing a messenger RNA enter from the outside may interpret this to be a virus. And so there's been extensive work in the field to understand what are the parameters that lead to the least immunogenic mRNA possible, and we think about it falling into 2 or 3 different categories. So one element of this that we didn't get into in this talk is that messenger RNAs have a 5' cap that is involved in enhancing translation or initiating translation, but it's also a mark of self and non-self recognition. And we think that having a proper cap that has what's called a cap-1 structure or a 2'-O-methyl group on the first sugar, it licenses the RNA as being a self-RNA within the cell. We've also found that we and others that chemical modification of the RNA can be important, and so in this experiment, when used a 5-methoxyuridine modified RNA, that also seems to reduce the immunogenicity. And then lastly, there seems to be a benefit from sequence optimization of the RNA. So the cell is recognizing, foreign and self-RNAs, and if you think about how a cell might recognize an RNA versus a DNA, it wouldn't be surprising that it might focus on uridines. And so what we found is that chemical modification of the uridines can be important to reduce immunogenicity, but also reducing the absolute number of uridines within the construct seems to be useful for achieving that purpose as well.
Thank you both. Time really just for a couple of final questions. What is the most effective method in your experience when to reduce off-target mutations, and how critical are these? We talked about off-targets earlier, but I think this is in a more practical setting. What tools, programs, or whatever strategies do you use to minimize off-target effects, and how far away are we to making this acceptable to be implemented in humans?
So Anton has alluded to this question earlier. So we usually pre-screen guide RNAs before we do experiments, just to choose or prioritize the guide RNAs with generally low off-target and likelihood. And in the meantime, we also use different deep sequencing or genome-wide off-target profile methods to compare the real off-target events in a cell culture-based assay. And then eventually we choose the candidate guide RNA with the lowest off-target effects and move into in vivo studies. So that's the typical research pipeline. And I'm sure there are also similar pipelines in the CRISPR industry or developing therapeutics, either ex vivo or in vivo. And there are already exciting development of ex vivo and gene-corrected and HSC cells being in clinical studies. So we're very close. It's really exciting field.
Yeah. Is there any software that you use to help in the design of your guides that you could recommend?
We use CRISPR and there's several CRISPR softwares we use. One is developed by Scott Wolf and Julie Zhu's group at UMass, and we're in close collaboration with them. There are groups predicting of targets or do genome-wide studies. Yeah.
Anton, anything you want to add to that? Anton may be muted.
Sorry, I was on mute. I would say that we're more of a provider rather than a user of CRISPR, and so I think I would defer to Wen's answer.
Fair enough. Is there, Pachi, another student, thanks again for your questions. Is there any possibility of Cas9 mRNA being silenced before it is translated?
Yeah, I defer this question to Anton, because he's
sure.
expert.
Yeah, I mean, so, the cell has methods for silencing RNAs that it finds to be pathogenic. And so some of the things that it's looking for, I already alluded to. That's the proper chemical modifications, the capping and modification of the RNA can, license it as a self-RNA. But one of the other things that is important is that during the in vitro transcription process, double-stranded RNA could be formed. And that can lead to an interferon response, which rapidly silences the RNA. Wen showed a slide at the beginning where he compared wild-type RNA to 5-methoxyuridine modified RNA. And the wild-type RNA did not work very well. And that's probably because of the presence of double-stranded RNA within the wild-type RNA that causes that interferon response. And one hypothesis is that these chemical modifications to the RNA are not necessarily improving the expression of the mRNA itself, but rather are silencing some of these double-stranded RNA responses. So basically, the modification is more important for silencing the double-stranded RNA rather than the single-stranded RNA. And so we think that also the purification and how you do that of the RNA can be very critical to whether or not you get silencing of the messenger RNA.
Okay, great. Well, I think we're going to have to bring matters to a close as we're at the top of the hour, and we have literally dozens of people still asking questions. Wen, I don't know if you would be so gracious as to give some of your time for people to contact you offline and respond to these individually. Would you be able to do that?
Absolutely.
Oh, that's so kind of you. So if we didn't get to your question, and clearly we didn't in dozens of cases, and it's really urgent, and you're respectful of Dr. Xue's time, you can email him directly. Maybe just put webinar question in your subject so he'll know the origin of the question. That would be wonderful. I really want to thank Wen Xue from UMass Medical School for a tremendous presentation, and Anton McCaffrey for joining the Q&A from TriLink. Thanks also to TriLink, obviously, for their sponsorship of today's webinar. And most of all, thanks to you for your attention and your flurry of great questions. This was the first in a three-part series of webinars from TriLink on different facets of CRISPR and genome editing. We're looking forward to hosting TJ Craddock in 2 weeks' time. And based on the extraordinary success of today's webinar, I really hope that TriLink will be persuaded to bring Wen Xue back for an encore performance in the coming months. We'll see if we can persuade him to do that. Hopefully, we'll see you all again for TJ's webinar in 2 weeks' time. But for now, and thanks also to Wen Xue for agreeing to take questions offline. That's very generous of him. All that remains for me to do is to say thank you again, and for everybody at GEN and the CRISPR Journal, including our friends at Workcast who hosted and provided the platform and technology for today's webinar. I'm Kevin Davis. Stay safe. Goodbye for now.